Good afternoon.
Good afternoon.
Could you please state your name and spell your entire name for the record?
My name is Yoli Brego, Jolibregu.
And what is your educational background?
I have a Bachelor of Science in Biology from Boston University and a Master of Science in Biomedical Forensic Sciences, also from Boston University.
And what is your occupation, ma'am?
I'm a forensic scientist too with the Massachusetts State Police Crime Laboratory in the DNA unit.
And how long have you been there?
Since June 2019.
Can you tell us your responsibilities as a forensic scientist too?
Sure. I perform forensic DNA analysis on evidence samples submitted to the lab. So my typical workday consists on processing those samples in the lab, doing the data interpretation, making comparisons with the reference samples, writing reports, testifying in court as needed. I also do administrative and technical reviews.
And what training have you had for this position?
So in addition to my education during my master's degree, I followed a forensic biology and forensic DNA analysis track. And then when I got hired, I was trained at the Massachusetts State Police, specifically in the DNA unit in forensic DNA analysis. The training consisted on lectures, reading of relevant literature, laboratory experience with practice samples, data interpretation, written and oral exams, followed by competency tests.
And did you pass those competency tests?
Yes.
And do you have ongoing training as well?
Yes, we have ongoing training. Every year we are required to take at least 8 hours of continuing education, which is offered in forms of seminars, webinar, workshops. We usually take more because there's more offered through the year. We are also required to read at least one scientific journal article relevant to our field.
And you have to take testing as well?
Yes, we have to take proficiency testing every year. They are taken twice a year.
And have you passed that proficiency testing?
Yes, I have passed the proficiency testing required throughout my employment. The last one is still in the grading process.
And have you testified in court about DNA?
Yes, I have.
Directing your attention to lab number 23-00596, did you do work on item 7-13.1, the sample from area A on the head of the hatchet?
Yes, I did.
And what work did you do on this item?
I performed the fourth step that is used in forensic DNA analysis, which is detection.
And how did you do the detection step of the DNA process?
So I took the samples from the analyst who had done previous work, Emily Oliver. I prepared those samples, mixing them with the required reagents. Then I loaded them in our instrument that does the detection, known as genetic analyzer, and I started the run. And that's where my role ended. After the run is done, the data is saved automatically in the computer and can be imported by the interpreting analyst for data interpretation.
Did you do the interpretation here?
No, I did not.
Who did the interpretation?
Salman Salim.
I have no further questions, Your Honor.
Any cross-examination of this witness?
Just a couple.
Go right ahead.
Thank you.
Yes.
Can I see that, Joe?
Yes, you did.
A couple of questions. You're familiar with the term in the context of DNA testing, allele, correct?
Correct, yes.
And the allele is, why don't you just tell the jury what?
The allele is a variant of a DNA fragment that is amplified during our third step. So it's a person has two alleles, one they inherit from the mother and one from the father.
Okay, and the allele then is the section of the DNA molecule that is then looked at once you've separated the molecule, and it's the part of the molecule that then gets amplified, correct?
Yes.
And so if you have an allele that repeats at a certain locus on the DNA molecule, then you may be a particular type at that particular location on the molecule, depending on how many repeating alleles you have.
It's how many repeats are within the allele. Okay. So the allele itself is the entire fragment, and within that alleles, there are we call them short tandem repeats. There are repeats, they repeat themselves different numbers for different individuals, and that's what it's assigned to an allele. How many times the fragment repeats itself within the allele.
And people can have the same repeats of alleles in the molecule, but generally speaking, they don't have the same number of repeating alleles across the molecule, various different loci.
Yes, correct.
And the loci is just plural for locus, right?
Yes, the loci is the location of that particular allele in the DNA molecule.
In the testing that was done in this case, you're using 26 different locations plus the sex area.
Correct, yes.
And that's a melogen.
Correct, yes.
Thank you.
Any redirect of this witness?
No.
The witness may step down.
Good afternoon.
Good afternoon.
Could you please give us your name and spell your name for the record?
My name is Karen Jacobson. That's KarinJacobsen.
And if I could just ask you to pull that microphone a little closer to you. Thank you.
Sure.
What is your occupation, ma'am?
I'm currently employed as a forensic scientist 2 within the DNA unit at the Massachusetts State Police Crime Laboratory.
And how long have you been there?
Since August of 2021.
And what are your duties?
I'm responsible for receiving items of evidence associated with known individuals, taking them through the 4-step DNA process, and making those DNA profiles available for use in comparisons.
And what is your educational background?
I have both a Bachelor's of Science in Forensic Science and a Master's of Science in Forensic Science from Bowling Green State University.
And do you have specialized training to be a DNA analyst?
Yes, I do.
And can you tell us about that training?
The training program within the DNA unit is at least 6 months long. It consists of lectures covering all of our protocols, literature readings covering current and historic methodologies, laboratory processing of at least 50 mock samples, and all of this is wrapped up with a competency test.
And did you pass your competency test?
Yes, I did.
And have you also had ongoing training?
Yes, I have.
And as part of your ongoing training, do you have proficiency testing?
Yes, I do.
And have you passed your proficiency training?
Yes, I have.
I want to ask you about lab number 23-00596. Did you do work on this case?
Yes, I did.
And what work did you do on this case?
The DNA testing process is 4 steps long. I performed the 2nd step and the 4th step for 2 items.
And what were those items?
These were items 13-1.1 and 13-2.1.
And what were those?
These were the DNA saliva collection kits for Anna Walshe and Brian Walshe, respectively.
Okay. So how did you do the step 2? That being the quantification step?
Yes.
Okay. Tell us about that, please.
In the 2nd step, quantification, I received a portion of the extract created in the 1st step. I applied a set of chemicals to it and put it on an instrument that goes through cycles of heating, cooling, and this produces an estimate of the amount of human and male DNA present within each sample.
And did you use equipment?
Yes, I did.
Okay. And what was that?
This is a 7500 real-time PCR machine.
And who had done the previous step?
The previous step was performed by Katarina Stashion.
Okay. And why didn't you do that step?
We batch our samples in the laboratory processing for the sake of efficiency.
Okay. And then when did you become involved again in this process for these samples?
I then performed the 4th step, separation and detection.
Okay. And can you please tell this jury how you did that 4th step?
So the same person, Katarina Stashion, performed the 3rd step. I received the product produced in the 3rd step from her. I applied another set of chemicals to it and put it on an instrument that separates each of the copies of the DNA by size. And then with the help of some special software, it turns it into a visual representation of the DNA profile.
And did you use any equipment?
Yes, I did.
And what equipment did you use?
This was a 3500 genetic analyzer.
Okay. And then what is the data next ready for in the process?
After the data is reviewed, it's ready for use in comparisons.
Okay. And who did the interpretation and comparison here?
In this case, it was Katarina Stashion as well.
And did you do any other work on these samples?
No, I did not.
I have no further questions.
Cross-exam? The witness may step down.
Thank you.
Good afternoon. Can you please state your name and spell your whole name for the record?
Good afternoon. My name is Katarina Stashion. First name is spelled Katarina. Last name is spelled Stashyn.
And what is your current occupation, ma'am?
I'm currently a criminalist in the biology section at the Washoe County Sheriff's Office Crime Lab located in Reno, Nevada.
And can you please detail your work history as it applies to forensic sciences?
I was previously a forensic scientist in the DNA unit at the Massachusetts State Police Crime Laboratory for about 3.5 years, from 2021 to earlier this year, 2025, when I got the opportunity to move back to Nevada to be closer to family and pursue a job still as a DNA analyst.
And what is your educational background?
I have a bachelor's and master's degree in biotechnology from the University of Nevada, Reno.
And do you have any specialized training to be a DNA analyst?
Yes, I received training at both the Massachusetts State Police Crime Lab and at my current job in Washoe County. In Massachusetts, I completed an approximately 1-year-long training program that consisted of reading scientific protocols, scientific literature, textbooks, all on the topics of forensic DNA analysis. I also took written exams and oral exams on those topics. I also practiced sets in the lab, both for laboratory processing and for DNA interpretation. Put together practice case files, wrote reports, did comparisons, and all of that training completed with a competency test, of which I passed. And then at Washoe County, I completed an approximately 6-month-long training course due to my experience, where I also did review of scientific literature, textbooks, protocols, practice sets, writing practice reports, performing comparisons or interpretation, etc. And that also completed with a competency test, of which I passed.
And have you also had proficiency testing?
Yes, I have also completed proficiency testing.
And have you passed those tests as well?
Yes, I have passed all competency and proficiency testing that I've taken so far.
And have you testified in Massachusetts about DNA?
Yes, I have.
So what is a single source DNA profile?
A single source DNA profile is a profile that originates from one person or one contributor. And you can identify this in a profile. Just by looking at the profile, you can see how many people are in there.
And what is a known standard?
A known standard is a sample that's taken directly from an individual. For example, a sample from somebody's mouth, a saliva swab, or a blood sample. It's taken directly from a person to be used for comparison purposes.
And how is a known standard developed into a DNA profile?
A known standard will go through the four-step laboratory process to result in a DNA profile that can then be used for comparisons.
And the four-step process is what?
The four-step laboratory process for DNA analysis is extraction, quantification, amplification, and detection.
So prior to doing any work, how do you prepare your work area?
So when you first get into the lab to do any of those steps, you're going to clean your lab bench. So that involves using a bleach spray to wipe down your area. You're going to clean all of the equipment, utensils, your pens, pipettes, any equipment that you're using. And then me as a person or any lab analyst, you'll be wearing a lab coat, a face mask, a hairnet. You're in full PPE, wearing gloves, changing gloves frequently.
Why do you clean your area and wear PPE?
We do this just to keep everything perfectly clean, or the cleanest that we can, just to help the integrity of the evidence and help produce the best possible profile.
Now I'm going to direct your attention to Massachusetts Case Lab 23-00596. Did you do work on this case?
Yes, I did.
And what did you do on the known standards in this case?
I performed the sample prep, the extraction, and the amplification on the known standards in this case.
And did you also do interpretation on this case?
Yes, I also reviewed the profiles that were obtained from that process and made sure they were able to be used for comparison purposes.
So I want to ask you about each of the things that you just told us about. So were you working on 13-1 and 13-2? Is that correct?
Correct.
And what were those?
Those were the known saliva standards for both Anna Walshe and Brian Walshe.
And so what was 13-1?
13-1 was the known saliva standard for Anna Walshe.
And what was 13-2?
13-2 was the known saliva standard for Brian Walshe.
And how did you do the sample prep for 13-1?
So both of these samples were known saliva samples, so a swab from the mouth, and I just cut up the swab and placed the cotton part of the swab into a tube, a labeled tube with those item numbers that I said.
Okay, so that's what you did for 13-1, and then did you give it another number?
So once I cut that swab off, it then became 13-1.1.
And so did you do the same thing for 13-2?
Yes, so then the cotton that got cut off of the whole swab became 13-2.1.
And then after you did that sample prep, what did you next do?
Then those samples went through the extraction step of the process.
And how did you do the extraction step?
So extraction consists of adding chemicals to the samples and going through heat variation to help break open the cells present in the sample and release the DNA that is present, and then that DNA is isolated.
And did you use equipment for that?
Yes.
What equipment?
An incubator was used for that heat variation that I mentioned, and an Easy One robot was used for the extraction part, meaning that after it sat in the incubator with the chemicals to break open the cells, a robot is used to isolate that DNA from the rest of the cellular debris left behind.
So you told us about sample prep and step 1. Did you do the next step, step 2?
No.
Why not?
It's just based on scheduling at the lab who was assigned to do what step that day.
And who did that next step?
The second step of quantification was performed by Jacobson.
Were you qualified to do all of the steps?
Yes.
And so what did you next do?
I next did the amplification step, that third step I mentioned.
And how did you perform the amplification step?
So amplification is the third step, and this is when we make millions and millions of copies of certain fragments of DNA. So this is done by, again, adding certain chemicals and going through heat cycles to make these many millions of copies of DNA, because we're usually working with a smaller amount of DNA. You need to make a lot of copies to make it visual and able to be detected.
Did you use equipment?
Yes.
What equipment?
We use what is called a thermal cycler, and this is the instrument that performs those heat cycles needed to make the copies.
So did you do the next step, the fourth step?
No.
Who did that?
Jacobson also performed the fourth step of detection.
So then what did you do next?
So after that full laboratory process was completed, I looked at the data that was produced from those 4 steps, and both items resulted with DNA profiles that I reviewed and made sure were acceptable to be used for DNA comparisons.
And could you visualize a single source?
Yes.
Okay. And did you generate a DNA profile for Anna Walshe?
Yes.
And did you generate a DNA profile for Brian Walshe?
Yes.
And were profile sources developed that were appropriate for comparison?
Yes, the profiles developed were acceptable to be used for comparisons.
And they were both single sources?
Yes.
What does appropriate for comparison mean?
When I'm saying appropriate for comparison, I mean that the profile is full, as in data is obtained at every location that was tested, and that it is single source.
And what do you do with the profiles?
That profile is then made into a text file that is made available for the analyst that's going to be doing the comparisons.
And were the profiles assigned a number to identify them?
Yes. So the numbers for the profiles are the same as their item number. So the text file profile for Anna Walshe also had the number of 13-1.1, and the text file profile for Brian Walshe was 13-2.1.
If I might have 1 second, Your Honor.
You may.
Nothing further, Your Honor.
Cross-exam. The witness may step down. Thank you.